In order for agriculture to contribute to making our society climate neutral, it's very important that we move carbon dioxide into the soil. And one of the ways we can do that is when we grow crops, but also cover crops. So in the autumn we have a window where we have no production of, or at least in some of our fields, there's no production of food. And when we have the cover crops, they both reduce nitrogen leaching, but they can also store more carbon into the soils via the photosynthesis. But we need to quantify and optimize this carbon input from the plants to the soil. And it's not only the roots and the leaves, it's also something called the phyllo and the rhizo deposition of carbon to the soil from the plants. Phyllo and rhizo deposition of carbon to the soil is easily degradable carbon compounds. And this pool is important because the microbes can easily digest and build microbial biomass from this pool. And the microbial biomass and necromass has been shown to be very important when it comes to the stabilization of the soil organic matter. We wanted to quantify this pool of carbon, and we wanted to test how the cropping system affect this phyllo and rhizo deposition of carbon to the soil. We tested in these fields, we tested the effect of long-term application of manure and the long-term use of cover crops, but also adding legumes to the mixture of cover crops. We found that the phyllo and rhizo deposition of carbon to the soil from the plant was about 3 to 10% of the total input of carbon from the plant, including the biomass. This seems like a small number, but since we know that this pool is important for the stabilization of carbon, it is actually a significant size of carbon. We found a high variability in this pool of phyllo and rhizo deposition when we looked at the treatments. So in the treatments with a high N availability, we found that the pool was lower, while in treatments with a low N availability, the pool of phyllo and rhizo deposition was relatively higher. And we don't know exactly why, but this is one of the things that we want to know more about in the future. We have also another study going on now where we look at this pool of phyllo and rhizo deposition down to 1 meter depth, because we can see that this pool is actually found all the way down to 1 meter depth at least. And it's important to get all of it into the calculations when we try to quantify these numbers.